Hi, my name is Chris Groves, and I'm Senior Manager and Site Lead for Cytometry, the Cytometry shared resource here at AstraZeneca in Gaithersburg, Maryland. Where our site is one of the lead or the core research sites within AstraZeneca globally. And our Gaithersburg, Maryland site, along with Gothenburg, Sweden, and Cambridge, United Kingdom, are the main research sites within AstraZeneca. So today, I would like to thank FLAS, and I would like to thank Beckman, and particularly Claire Rhodes and Marion Mott for inviting me to present some of the work that we've been doing with automation. So again, I'm in the cytometry lab, and what I want to talk to you today is about improving cytometry methods with the use of automation. So cytometry is a technique and a platform that allows for multi-parametric analysis of cells or particles. The technique is very heavily used in pharmaceutical industry, particularly where we're looking at antibodies or therapeutics. We can characterize many components of binding and proper function of our antibodies with cytometry. But cytometry, of course, is available throughout the world and is found in most major hospitals because one of the main things that cytometry does is aids in the diagnosis of leukemias and lymphomas, as well as HIV. So cytometry has been around for about 50 years. At AstraZeneca, we're testing the capabilities of the latest high content cytometers and really to get the most out of our technology. Our core team works to develop instrument quality assurance and quality control and performance tracking to control methods. And we utilize robotics and automation to improve the capabilities and reduce costs. And we advocate for informatics supported analytics reporting for quick, kind of simplified results. So what I'm going to talk to you today is about our work with utilization of robotics and automation. So cytometry is a useful technique or technology or platform that can be used from early discovery all the way through to manufacturing and preclinical and clinical studies. Cytometry assays are typically biomarker or bioassays that are used in clinical trials. Here at our site, we have a number of pieces of equipment, and we support about 150 users at our site that averages about 105 projects per year. So that's a lot, even for us. And really what I wanted to say is most of the projects that are conducted here at this site, not all, but most of them have a cytometry component to it. So it's a very heavily used platform in our drug discovery and development process here. So in our group, we have conventional, mass, and spectral cytometry. And as we, increase the number of parameters and the complexity that we have with newer, more kind of novel cytometer capabilities, we can see deeper and deeper into the immune system, and that we can identify cell subsets or interactions between cells that occur, and we can compare between health and disease to really find and discover, as well as kind of verify or validate targets for drug development. And then as we have, increased the number of parameters that we look at, we need better tools for informatics and data analysis. So traditional parameter by parameter plots get really complicated when you have 20 to 30 different parameters. So this is kind of where we are in the field of cytometry. Of course, there are those that go above, well beyond that with mass cytometry, and there are those that go well beyond that with spectral cytometry. But in general, we're on the 20 to 30 parameter range. But still, that data is complicated enough that we require dimensionality reducing techniques and algorithms to help us get to our results quicker. So where I'm coming from my perspective is automation, which you guys are well aware of, enables really new digital best practices. So one of the things that we're really striving for here is to standardize our methods and have means that we can implement to improve the quality of the data. And if we have some techniques that allow us to improve our productivity, then we can really advance where we are in terms of our capabilities as a research organization. So again, this is kind of what giving you an idea of what high content data sets look like. And when we use clustering programs and when we use dimensionality reduction techniques, we can see more broadly all the different subsets that are within our data. So here's an example of one of those kind of reference, one of those data dimensionality reduction and clustering algorithm data sets. So you can see here that we have a number of different unique clusters, and then we can use informatics to tell us the difference between those clusters in health and disease to help us identify cell subsets, in particular in this instance, that are relevant as a response to treatment or that are relevant in disease. One of the great things about standardizing our methods and utilizing robotics is that the software logs all the actions. So if there's a method that we're following and there's a problem, the robot won't move forward unless that error is corrected. And we know that when we're in the lab, sometimes we have to make these kind of quick decisions as we're conducting an experiment, and oftentimes those can be sources of error, or those can involve, when you make those decisions, if you don't keep notes of it and track it, then you're missing those bits in your methods, which kind of leads to being a little bit more difficult to reproduce. So the idea is if we can have robotic methods, then we can share those robotic methods or publish those robotic methods, then our data can be more impactful because people can easily reproduce it. And then when I look at my team, one of the reasons that I'm developing robotics and automation tools is really to improve the collaborative capability that my team in our kind of core or shared resource lab provides, and allow us to have greater productivity, as well as use the productivity that's inherent in automation to improve our capability and capacity. So, we're in a competitive field. I mean, science in general is competitive. But one of the things we always want to do is we want to evolve. We want to evolve our methods. We want to evolve our practices. And we want to be smarter, and we want to be faster than our competition. So as a core cytometry lab manager, and I would say most core cytometry managers try to evolve their capabilities and bring on new resources to their users. But what we really need to do is think kind of on a broader perspective about what that is. So when we have our resource, it's really like a business, right? So we need to kind of think a little bit more business oriented. And I get a lot of my kind of inspiration for business related information from the Harvard Business Review. So this is where kind of some of this information is coming from. So, when we're developing a method and developing a process, it also happens to be easier to convince other people if you have kind of a high level kind of business perspective and justification for the work that you're doing. And I'm going to go through our example of how we have utilized these kind of business skills or business tools to give us justification to work with robotics and automation. So really where it began for us was a cost saving exercise back in 2017. So back in 2017, the procurement team came to me and said, hey, we're spending a lot of money on reagents. And these reagents we know are being used primarily for flow cytometry, though they can be used for other techniques. Chris, being the expert in cytometry, what can you do to help us to try to initiate an initiative or best practices so that we can save some money? So what we did was we looked at the methods by which people utilize antibodies for cytometry. And we realized that people were not really titrating their reagents. So titrating your reagents is, starting with a high concentration and using lower and lower and lower concentrations, and then examining your data of antibody, and then examining your data and finding which one works the best or gives you the best signal with the lowest amount of background or noise. So what we ended up finding out was that people were just utilizing what manufacturers recommend for utilizing their reagents, which was 5 times, in some cases 5 times more than we actually really needed. And then we were thinking that if we titrated our antibodies properly, that we would get better results because of it. So what we ended up finding was that we were taking some shortcuts, and that when you add too much reagent, then sometimes we can have data resolution and accuracy errors. So a few years ago, we were working on, and we published a protocol to look at very rare antibody secreting cells. And what we found was when we used too much antibody, we couldn't see this kind of negative population, but everything was driven to be positive. So I'm just giving you a realistic example for what data looks like. And when you add too much antibody, everything appears to be in that kind of positive y-axis box, the big one. And that we lose, because they're very rare cells, we lose that kind of negative population. So this can happen. This was an example that I'm using to show you why antibody concentration and titrating and optimizing your experiments are really important. So then we were able to make those recommendations, and we were able to save some money. And then what we wanted to try to do was show that if we utilized automation systems to help us deliver those titration results, which we did, then what is the difference between manual and automated pipetting? So what we found was that we ran a number of tests on people's very complicated or very simple panels. And what we found was with the more complicated panels that were, 15 to 20 different reagents, when you're individually pipetting those reagents, there's a lot of variability that comes with manual pipetting. So we made some efforts to kind of quantitate what that variability was. And when we look at manual cocktailing, we're seeing really about anywhere between 20 and 40% CVs. And when we utilize robotic cocktailing, we can reduce those CVs to about 10%. And then when we further integrate with kind of full-on cell processing and reagent cocktailing, we can get our CVs around the 5% range, which is really outstanding. And if you figure that, I wouldn't say most clinical studies, but many clinical studies, and definitely a lot of research, is conducted like on a manual method, and is very similar to kind of what we're seeing here with very high CVs. And we believe that if we can lower the CVs and we can lower the variability of the work that comes out of our lab and our site, then we can improve not only the reproducibility, but we can improve, by lowering the variability, our ability to see things that might have been lost in noise. So automation, in our view, is something that's really important and is going to become even more important as we continue to advance our capabilities in the the high parameter and high dimensional cytometry. So one of the tools, and you saw a little picture of it earlier, is utilizing the LabSite Echo, or LabSite, which is Beckman now, Echo. And what the Echo is an acoustic fluid handling system that allows you to transfer liquid from a source plate that's got a special membrane on the bottom to a 384 or a 96 or whatever format plate you want to, and it shoots it kind of at a plate that's upside down. And it very accurately dispenses these nanoliter amounts. The one that we're using right now shoots 25 nanoliter droplets, but they have others that shoot smaller amounts of droplets. So we really got the idea from this, from the small molecule high throughput screening teams that exist in the UK and in Sweden. And they were using this technology for deposition of very trace amounts of small molecules and compounds when they're doing their compound screen. So we tried it out. It came here as a demo to our site, and in this kind of thing called the Future Lab here. And what we did was we said, okay, the Future Lab was saying, anybody want to try any of these things? And we said, sure, we'd love to try it. So we did, and we found that the Echo was incredibly quick at dispensing nanoliter volumes, so sub-microliter volumes of reagents. And we've done further studying to show the reproducibility, and I will show some of that data. I think I will show you some of that data. And then I will definitely talk about the flexibility for utilizing it in a variety of assays, and that's where we're going to go right now. But it's just so fast. And we're just amazed people by how quickly we can transfer, complex mixes of reagents. So here's an example of what we're typically doing when we do like a high parameter cytometry experiment. So we've got a number of time points in an animal model, a longitudinal animal model. And what that means is we've got an animal model that we're creating disease and we're treating them. And then what we're doing is we're sacrificing the animal and looking at some of the different organs or some of the different tissues of the animal to see what's happening, what happens in disease, and then comparing what happens when we add some treatments to it. So typically there are multiple time points in the study. And then what we do is we have kind of complicated 23 or 24 parameter panels, and we've got multiple mice and we've got multiple organs. So we've got a lot of plates that we're analyzing. And then we can do this on our instruments because we have kind of automated loaders. So for us, flow cytometry isn't about high throughput screening, but it's about high content analytics. So that's where the high parameter analysis gets us. So in this example, we had about 56 plates for this 4-week-long study. And for them to manually perform this on any given day, it would take them about 3 hours to do the pipetting for those 8 plates. When we did this on the Echo, it took less than 20 minutes. That is a huge and tremendous time savings. And that allows our scientists to spend that time making sure, not worrying that their experiment is working, but focusing on conducting the experiment properly, wherever we're not using robotics and automation. So this was just a fantastic example of the time savings. And this was manually one time. So when we look at the amount of savings per those 7 different time points, we're talking about half a week of time we were able to save our collaborator on this work, which is really getting and opening the eyes of the researchers that we collaborate with when we show them this type of capability. Another example of utilizing the Echo to transfer complex mixtures of reagents is an experiment that we were conducting where we had a number of different cytokines. So we had 10 different cytokines or 10 different growth factors. And what we wanted to do was put them together in every possible combination of 5 growth factors. So we had over 250 total combinations that we put together of those 10 different cytokines in 5s. And what we were trying to do here was to optimize cell growth conditions. So you can imagine we are working on regenerative medicine and we're working on cell therapy. So we're looking at ways that we can take cells and we can culture them and that we can optimally culture them so that they grow and differentiate and proliferate and have a certain function that we're interested in. So this work really was not possible if we were going to do this manually. It would have taken somebody probably a week to do all the pipetting. And when I talked to the investigators to try to, get some of this, they said, no, we can't. We're getting ready to publish this. So I'm not showing you the data and some of the readouts that they used. But this was just, they said that, if we have to do this again, and when we do this again, we're definitely going to use automation because it's just, I mean, we couldn't have done it any other way. So in this instance, we have automation and robotics making something that would normally have been impossible or we wouldn't have done because it was too complicated. Let the robot do that work for you. Another example of transferring small amounts of reagent was an effort that we were doing with PCR. So somebody came to us and said, "Hey, could I maybe utilize this system that you have, or fluid handling system, or automation? Can you support me to run? I've got half a dozen 384 well plates that I need to run PCR. I need to run PCR assay on." And actually, some of this, not this particular example, but this work was also applied to some of the work that that was done at our site and by one of the members of my team to develop a COVID PCR screen. So we potentially could utilize this, and we were doing it in our development stages to do COVID screening. So these are really, really powerful technique and a really powerful method that gives very consistent data and even allows us to reduce the amount amount of some of the very expensive reagents that we're using, primers and enzymes. So again, this is a huge and a tremendous time savings, but one of the things that we're trying to capture is how much money did we save, or how much money could we save if we develop this into kind of a more robust automated process. So we're using and utilizing just one piece, but in our kind of roadmap to kind of a more fully automated process, and I explained earlier that when we integrate robotic cell processing into the piece, we can reduce the variability and we can increase our capability. One of the other things, and one of the other benefits that we can have with an automated process is, you saw those pieces of equipment that we have in our lab. Those pieces of equipment are mainly used, and they're heavily used, but they're mainly used between about 11 o'clock and 7 o'clock at night. So when they're not used is first thing in the morning, and they're not used overnight. So for us to have the ability to have processing that occurs overnight, and that really enables us to run samples first thing in the morning, now we're tapping into improving our use and utilization of the resources that we have. So we can save money. We don't necessarily need to buy more analytical pieces of equipment. We just need to use it more efficiently. So one of the things here is I'm just showing you a diagram of utilizing different pieces of equipment. So the Echo there is, colored in kind of a yellow or maize yellow. And then we've got a Starlet that we're using for large volume fluid handling. And we've got our high-res arm, and we're utilizing the arm, and we've got centrifuges, and we've got plate washers in there. So basically what we're doing is mapping out and diagrammizing or diagrammatically showing you our workflow here. So one of the things that we're trying to do is develop a process that is our kind of, not good lab standards, but it's more or less like good lab practices. So we're AstraZeneca is, wanting us to adopt generalized procedures for best practices in the lab. So what we're trying to do here is create some documentation and qualification testing to enable us to develop this method in a kind of best practices in kind of qualified fashion. And then once we put this together, and once we're able to start utilizing a system, then we capture more of the metrics, which is time savings, reagent savings, and asset utilization savings. So these are things that we're going to build into the picture. And some of the things that were really difficult for us to get from our collaborators, we could definitely get the time savings, but it was more difficult to capture the cost savings from a reagent reduction perspective. So as we develop an assay, then we're going to qualify more assays. And then going forward, we need means to analyze the data in a little bit more robust and quick manner. So to qualify our assays, we really, I mean, we probably did a little bit more than what we needed to, but you know, we came up with you know, method qualification documentation that would be done kind of in a CLIA or more clinical setting. And again, this is in the style of good lab practices, but we don't require that for research. Even though we develop these, sorry, I'm in the lab, and when I'm not moving around, sometimes the lights turn off. And since I'm looking at myself right now, we've got the robot, and the robotic platform is sitting right behind me, and right here is the Echo that's tied into that system. So little plug, I am in the lab. We were working on COVID, and I told you a little bit about the PCR screening we were doing, but I was also using the flow cytometers here to help sort for antibody producing cells, which were used in manufacturing of our antibody clinical candidates. So we have product development documentation, verification, validation documentation, functional requirements and software and hardware requirements. And then we developed a plan to test, and we designed the plan, and then we're testing the plan, and that's where we are right now. So I wanted to show you some of the preliminary data that we have for our performance qualification. So what we were doing is utilizing different target volumes, and we were looking at the reproducibility, and we were looking at the linearity, and we were looking at the precision, and we were looking at the accuracy. So where we normally are with our cytometry antibody titrations or antibody amounts in our miniaturized assay, we're looking anywhere between 150 and 250 nanoliters. So when we designed this experiment, we wanted to capture the low end and kind of more the high end of where we normally go. And then we're showing you some of the data here showing you know, really outstanding linearity. And then we've done, you know, numerous days, and we've done different times throughout the day. And we're categorizing the individual data for accuracy and precision. And one of the things that I want you to really highlight here is the precision. Even though we had a little bit of variation at the high end, and we realized why we were getting some of that variation, is that between plates, when we were doing plates, we weren't filling everything back up. So we wanted to make sure that we filled that up. And when we did, we got much more reproducible data. And even before we did that, when we're measuring the precision, we're well under 2% CVs for precision, which is just incredible. It's outstanding, and something that we're really trying to show as an example of why automated pipetting or automated fluid handling is just much, much more accurate and much more reproducible than manual methods. So really for us, the Echo has been an essential tool in our ability to demonstrate how automation can assist in our day-to-day activities in a flow cytometry shared resource, and our day-to-day activities in the lab, preparing samples for utilizing cytometry, which again is a very highly utilized technique, platform, and you know, assay readout that we have here at our site. So really where we are going forward is to develop those analytics capabilities. And there are a couple of vendors here. You know, we talk about Omics and Astrolabe, and there's others. Those are two that we're particularly looking at right now, as well as internal analytics build, supported by our data science and AI team. What I really like to do is thank my team, in particular Rebecca Filippi, for the hard work that she put in for the greater part of 9 months so far this year. 10 months of the documentation, which goes into thoroughly exhaustive detail, and for her hard work in getting the automation data and automation experiments collected. Really, really want to thank Craig Lamison, who is our programmer. He's trained us on the Echo. He's supporting us on the Echo, and he's writing programs for us on the large fluid handling system. and we wouldn't have been able to do any of this stuff without him. Kelsey Newell is really providing us with some outstanding high-level support and justification for capabilities. I want to thank my boss, Tony Hess, for allowing me to do this, and her boss, Heren Wu, who's the head of our department. And I really want to thank also the collaborators, Jessica Araful and Sarah, whose data I showed you earlier today. And last but not least, our collaborators at CLAS Automation, who really were giving us the kind of documentation and kind of clinical orientation for our work and really helping us put that data together. Slide was put together by Carlos Aparicio and Cheryl Audici. That ends the seminar, and I'm happy to take any questions you have.